Hello everyone, my name is Eric. Thank you all for being here. Today I would like to talk about temperature stress causes host cell detachment in symbiotic Cnidarians, implications for coral bleaching. First, what are corals? Corals are Cnidaria animals in marine. They are marine invertebrates in the class Ancyloid of phylum Cnidaria. Corals consist of two layers, an outer ectoderm and an inner endoderm. Ectoderm and endoderm, along with the mesoderm, are the primary germ cell for any animals. What are coral reefs important? Coral reefs can provide habitat and shelters for many marine organisms. You can see this beautiful picture. There are many fish live in coral reefs. They support more species per unique area more than any marine environment. Coral reefs are sources of nitrogen and the other essential nutrients for marine food chains. Coral reefs assist in carbon and nitrogen fixing. They are some of the most valuable and diverse ecosystems on Earth. What problems are corals facing? Oil floating on the water's surface can be deposited directly on corals. And garbage or pollutants enter the sea and destroy corals' habitat. On this picture shows that corals in Great Barrier from 2012 to 2016. Coral bleaching in Great Barrier Reef is even worse than expected. It is estimated 70% of shallow sea corals have died because global climate changes rapidly. So what problems are corals facing? Seawater temperature has been rising gradually. We are going to focus on temperature stress causes coral bleaching. Before I talk about how to conduct experiment to provide insight to temperature stress causes coral bleaching, I want to talk about where do corals come from. Most tropical corals contain large population of symbiotic dinoflagellates. Symbiote rosenceller gives corals colorful pigments. The rosenceller are located in vacuoles within the host endoderm. Rosenceller can not only clean up the nitrogenous wastes, but also provide nutrients to corals. They are very important for corals. And this is rosenceller in corals. This is corals endoderm. Corals are sensitive. They would abandon rosenceller when seawater temperature changes. No matter seawater temperature is high or low, corals would release host cell to the seawater. Rosenceller could be released from the endoderm of Cnidarians by these steps. Step 1. Exocytosis of rosenceller from the host cell, resulting in the release of isolated algae. Step 2. Bleaching of the distal portion of the host cell, resulting in the rosenceller surrounded by vacuolar membranes. Step 3. Naturally occurring to cell death, and cell death because the factor external to the cell or tissue. Step 4. Detachment of host cell from the antigen. This is the host cell, and the host cell containing the complement cellular. These two pages shows that how to host cell detachment from chlorous antigen. Then I can explain how to experiment process. This is an animal collection. The scientific names are Aptesi potula and Polylopola lemmiconis. We refer them as anemone A and chlorophyll P. Both of them live in 1 meter depth. Their habitat temperature range annually from 21 to 27 degrees Celsius. This is Hawaii Institute of Marine Biology. All the experiments were being processed here, and the animal collected location is near here. Our objective. The research team wants to know mechanism of bleaching and morphology of cellular entity release. Both of them can be investigated experimentally. And then treatment, temperature treatment. This is cold stress, and the experiment and the control are shown in PowerPoint. And this is hot stress. All the experiments were being processing in the darkness. And the result. In your left panel, photomicrography of host cell release to seawater by anemone A in response to cold stress. And in your right panel is chlorophyll P. The photo clearly revealed host cell plasma membranes and vacuolar membranes surrounding the golden cellular, and surrounding the host cell nucleus, and surrounding the mitochondria. There's no photo shows that cell release after hot stress, because cell entities released after hot stress were too fresh to collect and handle. Discussion. Transient low and high temperature stress in darkness causes host cell detachment. And this phenomenon is primary mechanism of coral bleaching. The research team speculates that the cell release from antigen is caused by host cell adhesion dysfunction. Although they have described cellular entity released at temperature stress in darkness, there are the other underlying mechanisms, such as salinity and sedimentation, also evoke bleaching. This is the main point I want to make today. Corals are colorful because those cellular give them pigments. And when coral in a place start to bleach, they still can be saved if we improve environment immediately. Thank you for listening.
Okay. 9 minutes 30 seconds. Q&A. Andrew. Ed. Wally. When? Oh, sorry. Okay, in this order. Starting from Andrew.
Say center develop coral by temperature, and then you come to the conclusion, but I didn't see the data analysis.
Can you repeat this question?
You means I said the center it gives coral pigments, but I don't show the data in the conclusion. Those that are released by released from corals, right? Because of those ancillary, because host cell containing complete those ancillary, and then after temperature stress, they were released by coral and anemone, and showing microscope. So there is no data show this.
Okay. Second, Ed.
Can you show the sixth picture?
Six picture.
Six picture in the slide. Six coral. Slide 27.
27.
What is these pictures mean, or how do we explain this picture?
Okay. In the objective, we want to know morphology of the cellular entity release. And this picture is showing cellular entities. And in your left panel is anemone A, and in your right panel is coral P. Okay? This is in a microscopy.
Are you satisfied? More or less. Okay. Come back to this later. Third, Wally.
Sorry,
Can you go through the discussion? And the first, why is it just an environment? Is it just an environment?
Darkness. Can you repeat your question again?
Is it just an environment?
Is it?
Okay, sorry, it is not sure in a paper, but in this paper, all the experiments were being processing in the darkness. So when I make conclusion, I need to say this experience is processing in the darkness. Okay.
Do you get it?
This paper didn't show if this paper didn't show in light. Okay.
Okay. When?
Can you show me the page?
This is called stress.
Why you have
12 degrees Celsius. You say
and then you have 25 for 40 hours. Why is this time just no longer or shorter?
Can you repeat your question?
You control the temperature to low, from low to high. So I just want to know how you control this time length.
Oh, okay. Because we want to know if coral would release hot cell in a short time. Yes, in short time. We want to know coral would release hot cell in a short time. This is my conclusion. Transient low and high temperature. Transient means in short time. And this paper is beside the experiment.
Okay. One volunteer question. Any? Over there. Go ahead.
I'd like to know what is the range of temperature max? What's the close?
To say what is the range of?
Can we go? Can you say when cold or hot water can make
Here.
Yeah. What is the range of temperature?
I think the temperatures range from 21 to 27 degrees Celsius because the corals and the anemones habitat is like because the corals and anemones habitat temperature is from 21 to 27 degrees Celsius. So if the temperature is higher than 27 or lower than 21, the zooxanthellae would be released by coral.
Okay. My turn. I start with the bigger comments. I think the bigger comments would be you show too little data. If we look at the, you see you haven't even made your 10 minutes. And if you look at this paper, we see this one, we see this one, we didn't see this one, we see this one. However, that's the end of your presentation, right? But I don't know if you see this one, probably not. I think you should also show this one because this is the classical data. Because one of you asked, why didn't you analyze any data? Actually, you did, so you didn't show it. And this, in my opinion, is a very simple graph, and I'm sure you can explain it well. So in my opinion, for a short paper like this, you should introduce all the figures. Okay? And now you see the original figures in the paper. Now let's go back to your presentation. Okay. So if I go through your presentation, I think your introduction part, again, I think the basic information is nicely provided. So what are corals? And the pronunciation is also correct, Cnidaria. Okay, and here you have, these are all well done. Okay, where do corals cover? You see the error? You see the error? Okay, where does? Hmm?
But corals,
It's
the sweaters.
the objective is color, okay?
Okay.
And then during your, I think you memorize your script very well. And therefore, I highly recommend that you should look at the audience more often. I have the feeling you already spoke fluent. In that sense, you should be more relaxed, because sometimes you were reading like looking there, and then I know you are trying to memorize your line. Huh? Okay. And then you see this figure in the paper, and did you add the color yourself?
No.
Where did you get the color?
In the other in this picture, I
From the website.
In the other paper.
Okay. However, I like this color version. However, I think based on my rough understanding of the paper, the way you present it is more like these are different steps of Zuzendila excretion. However, based on the paper, it's actually different theories. 不同的理论，不是不同的阶段。Okay? So what the author trying to do is they want to see which theory is correct. Okay? So it's not different steps because you say as if it's different steps. And then here, can anybody tell me what's the correct way to write down? Because many of you have biology paper. What's the correct way to write down the Latin name of an organism? Here you see errors. And there was error in the previous talk.
Dots.
No dots. What's the error there? Many of you like biology, then you should know this basic knowledge. What's the error here? Can anybody tell me? Do you know your error? There should be no comma here. Comma means this one. 逗号, no comma. No. And then the second time you mention the organism, you can put a dot. Procella. Okay? Dot. A 一点. Procella. And the Latin name has to be italicized. 简体化. Okay? And then what I like this presentation most is there's a very clear objective slide telling you what he wants to achieve in this presentation. I highly encourage you to do this. Okay. And then when talking about the key finding of this paper, what was missing is you did not use your pointer well enough. You have these abbreviations, right? You should point to it very clearly. Very stable, very clearly. Okay? How do you pronounce this one? Anybody? Mitochondria. Mitochondria. Okay. And then there's a very common, I would say, flaw in your presentation. In your discussion, you start with these two sentences, right? You sort of read through the sentence. However, after these two sentences, when you go back to your video, you will see you talk a lot about something else,
Yes.
like salinity things, etc., etc. When you talk like that, then all the audience is sort of lost. They don't know what you are talking about.
Okay.
So you should at least give some keywords. Do not say a very long script without any keywords shown on the slides.
Okay.
Okay. And then another thing I would like to warn you is never do a font like this. This is 标楷体, okay?
Oh.
Oh. Please use the font